Welcome, everyone. Thanks for thanks for joining us today. For those of you who are joining live in Zoom, we are going to have a poll that pops up, and we are interested in getting a sense of how familiar you are with phenology. Have you never heard the term phenology before this? Do you know a little bit about phenology? Are you already familiar with phenology, or are you already a phenology expert? While people are joining, we're just going to leave this poll up. So take a moment, give us your answer. We'll share the results in a moment. And in the meantime, while you're all answering, I'll go through a little bit of housekeeping. So please use the Q&A to ask questions. We've got a lot of folks joining, so the Q&A will be how you can get your questions answered. We're going to have a couple other live polls for you to answer, and we'll also be sharing some useful links in chat, including directing you to how you can add annotations in iNaturalist 2, which will be the second half of this presentation today. And yes, don't worry, this is being recorded, and we will send you a copy of the recording as soon as possible. So you'll be able to go back, rewatch things if you want. Don't worry, it's being recorded. So welcome, everyone. I'm so pleased that you all can join us to learn more about phenology and how you can explore and contribute to the understanding of plant cycles through iNaturalist. So let's see what the results of the poll are. For those of you who've been attending live in Zoom, we've got so many people new to phenology. 30% of you have never heard the term phenology before this. That is awesome. 43% of you know a little bit about phenology. That is awesome too. Then we've got 25% of folks who are familiar with phenology, and 2% of you are already phenology experts. So we've got a great spectrum of attendees today. This is awesome. I'm especially excited about those of you who are brand new to phenology who are joining. And even if you're already familiar, I hope that you'll learn something exciting today too. So my name is Dr. Carrie Seltzer. I'm the head of engagement for iNaturalist, and I'm so pleased to be joined today by Erin Grady, as well as Dr. Vijay Barve and Dr. Jeremey Yoder. Erin's going to give us an overview of plant phenology, help teach us what it's all about, and Dr. Vijay Barve and Dr. Jeremey Yoder are going to be telling us about some of the research that they've done with phenology using observations from iNaturalist. I'm also especially grateful to everyone behind the scenes who's made this possible. We've got my colleagues Tony Iwani, Thea Scaff, and Allison Young, who are going to be helping to answer questions you have, as well as dropping links in chat. And thank you to Walker Irving, who's running the Zoom and has been answering some of your questions as well. And we're grateful to the support from the U.S. National Science Foundation under a collaborative research grant that has supported a project called Phenobase that iNaturalist is involved in, and you'll hear a little bit more about that and how it's helping make phenology data from iNaturalist and many other programs as well more widely available. I also want to acknowledge the almost 3 million people who have added plant observations to iNaturalist already, including, I hope, many of you, as well as the more than 200,000 people who have shared their expertise in helping identify those plants. We couldn't do this without all of you, so thank you. And now I'm happy to turn it over to Erin Grady to give us an overview of what phenology is all about.
All right. Hi, everyone. Thank you so much for joining us today. We are just so excited that you're interested in learning about and participating in our phenology work. My name is Erin Grady, and I'm a master's student at the Florida Museum of Natural History, as well as a member of the Phenobase team. So I spend a lot of my time thinking about and working with phenology. And I see that some of you are already familiar with phenology, which is great. And I'm so excited for you to hear from Vijay and Jeremy about all of the cool phenology work that they've been doing. But I'm also so excited to see that many of you are not as familiar with phenology. And so I'm going to give a brief overview about what is phenology, why do we care about phenology, and how do we collect phenology data. So when I say phenology, I'm referring to the timing of seasonal changes in plants and animals. So for example, bird migration, the changing color of leaves in the fall, plant flowering and fruiting, insect activity and emergence. All of these things are examples of phenological events, so they happen at certain times of year. And the thing that I think is really cool about phenology is that it's one of the first and main ways that we as people engage with the natural world around us. So we notice patterns and we pay attention to seasonal changes, and we get excited for these changes. But sometimes we don't think about why these changes are so important. And actually, the timing of these biological events upholds the structure and function of ecosystems. And many processes rely on timing. So let's take plant-pollinator relationships as an example, and I'll use this throughout. Pollinators rely on plants for important nutritional resources like pollen and nectar, and then plants rely on pollinators to successfully reproduce. And so this relationship only works if the plant is flowering at the same time the insects that rely on them are active. And so you can see how this relationship is structured based on timing. And this is just one example of many processes that phenology is important for. Many species interactions, reproduction, food webs, carbon cycling, competition. These are all examples of processes that rely on phenology, and they uphold the structure and function of ecosystems. Now, when this gets a little bit tricky is when we start thinking about our changing climate. And that's because phenological events are triggered by environmental cues like temperature, precipitation, day length. And with human-induced climate change, we're starting to see rising global temperatures and increased extreme weather events. So we have these hot periods, cold periods, and overall rising temperatures that are abnormal. And these abnormal environmental conditions are causing the shift of phenological windows. So for example, we're seeing a lot of plant species are moving their flowering windows earlier in the year based on these warming temperatures. And these shifts in phenological windows can have big ecosystem problems. So if we take that plant-pollinator relationship again, if that plant shifts its flowering window to earlier in the year and it misses when that insect is active, suddenly this mutualistic relationship doesn't work, right? So that plant can't successfully reproduce and the insects don't get the nutritional resources that they need. And so this is just one example of a process that's dictated by phenology that gets really impacted by climate change. But this is relevant to all of those processes that we just talked about. But not only is phenology important for ecosystems and ecosystem processes, but it actually is really important for us too, and it touches a lot of parts of our lives. One of the main examples of this is agriculture. So it's important for us to understand when the crops that we rely on are going to be available, and when the insects that pollinate some of these crops are going to be active. And then how is this changing based on climate change? There are also public health implications of phenology. About 1 in 4 people have seasonal allergies, and understanding when that plant is flowering and when the plant is releasing pollen is really useful to understanding when your allergy season is going to be. And then is your allergy season expanding or shifting because of climate change? A lot of tourism relies on phenology. We have towns that prepare for these different events, like the blooming of cherry blossoms or fall foliage, and people plan their trips based on when we predict these events will occur. So these are many examples of ways that phenology touches our lives. But I think the most important one is that phenology is an ecosystem health indicator. And what I mean when I say that is when we see these shifts in phenology, that's an indicator to us that that ecosystem is starting to be impacted by climate change. And so it's really in our best interest to monitor phenology and understand how it's changing so we can understand the impacts of climate change on our world. So let's talk about how we monitor phenology. The cool thing is that people have been interested in tracking phenology for thousands of years, and indigenous peoples were the first phenologists. They paid attention to the timing of the plants and animals around them, And they use this understanding to inform things like resource management, when to plant, when to harvest, when to hunt and gather. The first written records of phenology are thought to be from China, and we also have records from Japan from a couple thousand years ago, marking when those cherry blossoms were blooming. We also have record of early Western naturalists who recorded phenology in their local environments. So we actually have very detailed records from hundreds of years ago, but they're often from certain locations or for certain species. So where do we get phenology information now? And there's three main ways that I'll touch on here. The first and the most traditional is through phenology monitoring programs. And these exist globally, and they consist of researchers or volunteers repeatedly going to the same sites and recording phenology information about either plant individuals, plant communities, plant species. And we get really detailed information from these monitoring programs, but they're often in very specific locations and for a certain list of species of interest. Now, the second way we get phenology data, and the focus of our webinar today, is through community science platforms like iNaturalist. And actually, every plant photo that you upload to iNaturalist contains important phenology information. So for example, if we look at this chocolate lily observation, this tells us that a chocolate lily was flowering on that day in that location. And that's a really valuable data point for researchers interested in phenology. And you might have participated in other community science programs like Nature's Notebook, which is run by the National Phenology Network. And that's a cool one because it's kind of in the middle here. It's a community science program, but it has a more structured approach. So you can go back to the same sites and choose from their species list. The unique thing about iNaturalist and how we get phenology information from iNaturalist is that everything is a one-time observation. So you can make an observation of any species in any location at any time, and that observation will contain phenology information that can be useful. And because of this flexibility, we have millions of plant observations that contain phenology information. But the tricky thing is that we haven't quite unlocked this phenology information yet. So if we look at this chocolate lily, you and I know that that's flowering. But until we annotate it and write that it has been flowering, it can't be used in research. And so that's where these phenology annotations come in. And you can help make these phenology annotations on iNaturalist and help unlock this phenology information. And Carrie's going to do a great live demo in a bit to talk about how exactly you can do this and what different annotations you can make. But I also want to emphasize that continuing to make new iNaturalist observations and also helping to ID those observations is just as important in increasing our phenology knowledge. And so we are just so grateful about your efforts and that you're excited to collect data and make annotations. Okay, so we've talked about phenology monitoring programs and community science initiatives. Both of them are ways that we could get phenology data. The last one I'm going to touch on is historical collections. And we get these when researchers go out into the field, collect a plant, bring it back, press it, and mount it on paper. And these are stored in something called an herbarium, which is essentially a plant library. And the reason that these collections are so important is that they often can show us what phenology looked like back in time. And they outdate some of these community science programs and monitoring programs that are a little more recent. And so historical collections can give us that really important information on what phenology looked like in the past, and we can compare it to the present to understand how it's changed. So we have all of these different kinds of phenology data, from monitoring programs, community science platforms, historical collections. They have different levels of detail, different species, they're in different locations. So how do we work with these data? And this is where Phenobase comes in. So we are a National Science Foundation funded initiative, and our goal is to take data from all of these different places, combine it, standardize it, and then produce a database that researchers can go to download high quality global phenology data. And a big part of what we do is we train machine learning models to be able to look at images and classify the phenology stage in those images. And the cool thing is that we are actually using your phenology annotations to do this, to train our models. So we take your annotations, we pass them to our model, and we use them to help our model identify patterns. Like you and I understand what a flower looks like and what a fruit looks like, but we need to help our computer understand those things. And so once our computer learns to identify these patterns and identify these different plant phases, We can then pass unannotated images to our models, and it can help annotate those images that haven't been quite unlocked yet. So every time you make a phenology annotation, not only can it be used by researchers like Vijay and Jeremey, but we actually use it to help improve our models. And so we just so appreciate these phenology annotations. It also helps track as iNaturalist continues to grow so that we can continue unlocking that phenology data. And the last thing I want to emphasize here is that because of Phenobase, but really because of iNaturalist and because of you, for the first time, we're able to get this near global view of phenology. So we have phenology data in all of these different locations for different species, different years. And this is really novel and exciting because it allows us to do research that has never been done before about how climate change is impacting our world and how phenology is shifting. And so we're just so grateful because this wouldn't be possible without people on the ground making observations and people making these phenology annotations. And I'm so excited for you because next up you get to hear from two really awesome researchers. They use iNaturalist to do their phenology work. And then Carrie is going to give a live demo of how you can make those phenology annotations we talked about. So again, thank you so much for being here. I hope that for those of you who weren't as familiar with phenology, you feel like you have a better sense of what phenology is and why we care about it and how we collect phenology information. And with that, I will take questions.
Thank you so much, Erin. One of the questions that we got a lot is, can I or should I make repeat observations of the same plant over time, or is that bad?
That's a great question. The short answer is that's great. And we are so excited that you are excited about taking these phenology annotations. The one thing that I want to note is that make sure you make a new observation every time you go to that repeat place. So make sure you're not adding images to the same observation. Every time you go, you make a new observation. The other thing I want to note is that when researchers use iNaturalist observations for phenology, they usually do it at a species level or higher and not at an individual level, because there's no way for them to know across observations if that's the same individual or not. And so what you're doing by making those repeat observations is you're helping them understand how the phenology of that species in that location is changing. You can also, if you're interested in doing it at an individual level, you could do it on your own project, but in general, we're using it at a species level or higher. So short answer, yes, make a new observation every time.
Awesome. Thanks, Erin. We're going to pop up another quick poll for you while you answer this next question. So many people asked, how can they make the most useful observations for understanding plant phenology? What do they need to make sure to include?
That's also a great question. I think the most important thing is for that plant individual, make sure you're capturing in your photos every unique phenology phase occurring on that plant. So for example, if you have a plant that has both fruits and flowers, make sure that both of those are in the photos for that observation. And the other thing is if make sure you take photos up close and with good quality so that people can make those annotations. If you take a photo far away, make sure if you can walk up closer and get those photos of flowers and fruits so that we can identify the phenology phase.
Cool. So different parts and different scales, especially getting that close up, but sometimes getting the far away perspective too. That's
Yeah.
great. Thanks. Let's check the results of this poll. We can go one more slide and see what the answers that came in are. Um, Erin, can you go to the next slide?
Yes.
Cool. Okay, so thank you all for taking this poll. The question is, have you contributed to phenology research already? For example, by making iNaturalist observations or other phenology programs. 25% of you said no, not yet. 23% of you said maybe. 49% of you said yes, definitely. And 3% of you said yes, and I've written it. So we know we've got some phenology researchers and experts in the audience. That's awesome. So next we are gonna move to research presentations. First from Dr. Vijay Barve, who is going to tell us about some research that he did with iNaturalist observations from the genus Yucca. Vijay, can you briefly introduce yourself and tell us about your research?
Yes. Hello, everyone. My name is Vijay Barve. I am currently with the Natural History Museum of Los Angeles, and I've been interested in biodiversity documentation for a long time. So I'm going to talk about this study that we conducted. We tried to assess phenology with this group of plants called Yucca. So yucca is a group of perennial shrubs and trees. It has a wide variety of plants in that. All of them are native to Americas, and about 50 species of yucca uh, are there in the uh, next one. Yeah, so when we started formulating this research, our objectives were we were looking at developing methods and best practices for using iNaturalist. Basically, how can we extract data from iNaturalist and analyze for phenology? Uh, we decided to use yucca flowering as the case study and learn what influences actually yucca phenology. What what are the drivers behind these phenological patterns, and we also wanted to determine with yucca whether the flowering is out of its typical season or and then what are the drivers behind it. So the data sources we considered was mostly all the yucca observations at that point. So there were about 30,000 observations when we did this study, and there were 36 species covered in the in our nature list at that point with observations, and there were about 8,000, more than 8,000 observers who had contributed by then, and there was six species that we considered deciding on. We wanted at least more than 100 photographs from iNaturalist so that we can actually look at the flowering patterns. So these are the iNaturalist numbers, and these are the NPN numbers for the same plants that were uh, available at the time of analysis. So what we did was we basically mapped the range of the plants from literature, and then we plotted the iNaturalist and NPN observations on that to see how best it is fitting. Next one. And then we analyze all the flowering uh, of this data set. So for every year, all the gray boxes you see are the observation of yucca in that week, and the red color shows that the flowering was happening at that point. And depending on how dark the color is, number of records were found. So for the red color. And then we plotted these records, special distribution of flowering, and we basically decided to uh, plot in two different maps. One is the normal flowering time, which is for this particular species is March to July, and then the other one is the abnormal flowering time, which was for this particular species, August to February. And then if you look at these maps, you can see that the yellow color normal flowering is fairly distributed across, but abnormal flowering in this case was in a particular region. And then when we explored that further, we realized that it was due to a kind of untimely rain in that region. Next one. So all these methodology and results are available in this particular research publication. If you want to find it, it's open access available. So um, you can take a look at it. Thank you.
Awesome. Thank you so much, Vijay. It's really neat to hear how iNaturalist observations allowed you to catch these unusual flowering events that happened in some particular years and see how they were distributed spatially as well. So we'll answer a few questions after the next presentation from Dr. Jeremey Yoder, who works also on yuccas, also in the same part of the world. But I think it's cool to see how you can take the same data and do something quite different with it. Jeremey, are you ready? See you. There
Hello.
you,
Yes, I seem to be muted.
you're unmuted now.
Sorry. I don't have I'm not able to turn on video.
We can at least see your picture on Zoom. So go ahead.
All right. Well, hi everyone. I'm Jeremey Yoder. I'm with California State University Northridge, and yes, I study Joshua trees, which are a couple of different species of yucca. This is a particularly nice stand. Um, uh, And yeah, so next slide, please. Joshua trees are two species of yucca, Yucca brevifolia and Yucca jageriana, distributed across California, Nevada, Utah, and Arizona. And I study primarily their pollination interaction and their evolutionary history. But one of the things I've come to realize is they're a really great case of um, examining something that we often want to know about species we study or we want to protect. Next slide, please. And that's that when we are trying to study or protect a species, we need to know where it lives, what kinds of habitats it needs, but also how it's doing in those habitats. And Joshua trees encounter a lot of variation in conditions. This is a population on the outskirts of Death Valley. It's very low density. The trees are not doing very much in this spring moment. But not that far away as the crow flies in the eastern Sierras, we have a very different kind of population. Lots of really elaborated trees. This is higher elevation and getting a little more precipitation. It's basically on the Pacific Crest Trail, and you can see this tree is really, really flowering. This is not, I think, the same year. But Joshua trees don't flower every year. And so the phenology data in iNaturalist gives us a handle on how they're experiencing the conditions year to year. So I worked with an undergrad researcher, Ana Karina Andrade here, to go through Joshua tree records on iNaturalist and annotate their phenology. And we came up with thousands of records, over 10,000 from more than 1,600 unique contributors. And you can see these are unevenly distributed across the range of where Joshua trees grow, the green shaded areas here, and in time too. We have a lot more contributors to iNaturalist in 2022 than we did in 2008. But we use this data in a machine learning model relating weather to flowering. So we could ask what weather conditions predicted flowering of Joshua trees in a given year. And that's really neat because it means that we can go to times where we have weather data but no direct observations and predict what the trees are doing. So we're calling this method temporal analysis of reproduction distributed in space, which does spell out TARDIS because it is a kind of a time machine for understanding how Joshua trees have experienced past weather conditions. So we can go to historical weather records and ask how likely it was that Joshua trees flowered. Here's 1905, here's 1936, the year that Joshua Tree National Monument was founded. 1994, or so 1994 and 2005, that's when the National Monument became a national park, and the year before my first field season working with Joshua trees. And these hindcast predictions are just predictions, but we went back and compared them to historical records that are independent of the iNaturalist data, field notes from a collaborator, preserved herbarium specimens, even newspaper stories. This is a 1924 LA Times article about a really good Joshua tree flowering event. The TARDIS model predicted that. So with that, history that we've reconstructed, we can summarize across more than 100 years, more than 120 years of history to see how conditions have affected Joshua trees over that period. And on average, the model predicts that they flower about once every 4 years. It also predicts that they're flowering a little bit more frequently. In the early 20th century, they flowered about once every 5 years. More recently, they flower about once every 4 years. And that reflects bigger changes year to year in rainfall, which is what the trees respond to. So it's not actually necessarily good for the trees. But our current plans for this work, we are extending the TARDIS approach to understand Joshua tree pollinator activities. They only make fruit when yucca moths pollinate them. So observations of trees with fruit tell us where the moths have been. So we can use that data in a very similar way. We have a project right now collecting observations to do that. And we're working on extending this method to a whole bunch of other species with different kinds of reproductive biology than Joshua trees. So we're excited for future applications. And so you can find the paper that describes all of this in Ecology Letters. It's open access. And I have a blog post explainer that talks about it in a little less technical terminology.
Awesome. Thank you so much, Jeremy. I just want to highlight that if you made a Joshua tree observation sometime before 2020 and posted it to iNaturalist, it was probably used for both Vijay's research as well as Jeremy's research. And so that's super cool. And I hope you find that exciting. And there's so much more happening, and we're excited to figure out how to tell a lot more of these research stories of all the cool things that are coming out of iNaturalist, because right now it's hard to know. So something that I think you both touched on a little bit, but we got several questions in this vein. Is phenology only affected by weather, or like what else can influence phenology and the timing of things? Vijay?
Well,
Go ahead, Jeremy.
I could say it definitely varies from species to species. Um, uh, but one of the most consistent triggers for plants for the timing of flowering is how many days they experience with temperatures above a certain threshold. So for plants in especially temperate climates, they're trying to avoid getting frostbitten when they flower. They are waiting for it to warm up enough that they're pretty sure that when they flower, they don't experience frost, right?
Yeah,
And also a few events like the fire or untimely rain triggers slightly different patterns in phenology. So that is another factor which is localized but definitely observed.
Yeah, I also wanted to mention by coincidence, this was absolutely not intentional, but both Vijay and Jeremy are from the Los Angeles area, which is feeling the effects of devastating fires recently. And many people have also asked questions about what iNaturalist observations can tell us about plants after fire.
Well, INAT
I
observations.
think,
Oh,
yeah, go ahead.
I was going to say INAT observations are great for tracking the recovery of the natural community after fire. And we're in a chaparral environment, most of LA. So there are a lot of fire adapted species that are ready to go as soon as the fire has passed. And we do expect regrowth to get started already.
That's great. Anything to add to that, Vijay?
Yeah, I think it's important as soon as people get chance after the fire, start reporting what is regenerating first and how fast they kind of start researching is also very interesting for future studies.
I guess I would say you do want to make sure you're waiting until it's safe to go into the
Yes,
the
yes.
burned areas. I think many of them are going to be closed for public access even after the fires are fully out.
Mhm.
But as soon as they reopen, observations will be helpful.
That's great. Great reminder that we never know what the future holds, and making observations today, make them today, because a fire could happen in the future. That historic information is important and yeah, helps us understand what recovery might look like. So we're going to have another poll. So while I switch to sharing my screen, those of you who are in Zoom, we've got a quick question about whether or not you have added phenology annotations before. So please fill that out quickly. And I am going to start sharing my screen here, and we're in the sort of live demo section. Again, this is being recorded. We'll send you a copy of the recording later today, so you'll be able to go back and refer to this. Pause it, rewind, watch some parts of it again. I did make my cursor extra big so that hopefully you can see it easily, but I'll keep it out of the way for now. So let's see the results of that poll. And so far, we've got 65% of you have not added phenology annotations before today. That's awesome. Our goal is to turn those no's into yeses. We've got 22% of you who have. That's awesome. I hope you'll learn something new. And then we've got 13% of you who are not sure. Awesome. So let's get down to it. I want to emphasize here that we really need your help and you can really make a difference. Currently, iNaturalist has 94 million observations of flowering plants, and only about 10% of those have any annotations about flowers or fruits. For vascular plants, we've got almost 100 million observations, and only a tiny fraction of those have annotations about leaves. There's only a few hundred people on any given day who are adding these, and there's almost 5,000 people watching this webinar. So we can all chip in and we can all collectively make a difference here. When it comes to plants, there's a little bit of sort of framework that I think helps understand what's possible here and why certain annotations are available for different plants and not others. Non-vascular plants are things that basically can't get very tall because they can't move water very far. So this is things like mosses and liverworts. They stay very close to the ground. Right now, we don't really have any annotations for mosses or liverworts. We may in the future. But most plants are vascular plants, and vascular plants have some kind of leaves, even if they're not very prominent, even if they actually look like needles, which are just specialized leaves. And so conifers and ferns are examples of vascular plants, and you can add leaf annotations to those plants on iNaturalist, as well as flowering plants, which are by far the most diverse group of plants on Earth. And for flowering plants, this is where you can add annotations of flowers and fruit. So here's an example of what this looks like. I'm using the genus Prunus here, which is very economically important. Erin mentioned one of its important elements, which is that these are very beautiful. Ornamental cherry trees have been used in phenology, in recording phenology. There's often festivals associated with them. There's over 1,000 years of records of when cherry trees bloom in Japan. So this is an important phenological genus. It includes things that we love to eat, like apricots and peaches and cherries and plums and almonds. And so that's why I'm highlighting this particular genus here. Over here, we've got an example of when you should add an annotation of no flowers or fruits. If you can only see leaves, for example, or if you only see bare branches, then no flowers, fruits, or seeds are visible. I want to emphasize that this is based on the evidence that's shared as photos in the observation. So it could be that there's flowers or fruits somewhere else, but if it's not captured in the observation, we're going to make the annotation based on the evidence provided. For this apricot here, we've got an example of flower buds. So in this case, this is all flower buds that are closed up. They haven't opened yet. But if there's at least one closed flower visible in the photos, you can add the flower buds annotation. For flowers, there should be at least one open flower visible, which is clearly visible in this photo of a Chinese wild peach. And lastly, for fruits or seeds, there should be at least one fruit or seed visible. We're not asking you to distinguish between ripe or unripe fruits, since that can be very difficult in some species. So just if you see fruit or seeds, you can annotate it as fruits or seeds. For leaves, we're going to use a different example genus. The genus Acer includes the maples, which are found on most continents, introduced in some areas, but they're especially noteworthy for their brilliant autumn-colored leaves. So we have first an example of no live leaves. You can see there is a leaf in this photo, but it's a dead leaf. There's snow, there's a twig that's hanging on to an old leaf. So this is an example of when you would indicate no live leaves. For breaking leaf buds, I'll say this is absolutely the trickiest phase to identify. And so if this is not something that you're already familiar with, you can feel free to skip it. You can just never add any breaking leaf bud annotations, and that is totally fine. You can focus on the easy things like no live leaves or green leaves or colored leaves. So most plant observations on iNaturalist are probably going to get a green leaf annotation. And it is important to note that this is intended for sort of normal leaf color. So in some plants, the normal color is reddish, still intended to be green, normal colored leaves. Colored leaves is really intended to be an indicator for late season, so in the autumn in temperate areas, or drought coloration, which is beautifully illustrated by this Italian maple. In chat, we'll put a link to where you can find the definitions to these annotations in the iNat help documentation, so you can refer to those in the future. We'll also share a link to these slides if you want to refer back to them or perhaps use them in a presentation that you give yourself. So now I'm going to switch to sharing my screen showing the iNaturalist website. So I'll say first that you'll need to be logged in on the iNaturalist website. Many of you might have only so far used the mobile apps. You can add annotations in the Android app, not in the iOS app yet. So we're going to focus here on the website. And you'll need to log in to add your annotation. I've pulled up an example of a prunus observation that I made. You can see there are both flowers and buds visible. There's different photos. This one's entirely showing buds, and this one's a sort of not as clear view, but thankfully I've got these other photos too. And this is what we call the observation detail page. If you scroll down a little further here, there's a section called annotations. It might be collapsed, in which case you can just click to open it. And in this case here, someone else, TSN here, has both added an identification and added annotations here for flower buds and flowers. So this is a great example of how someone else can help annotate your observations, and you can help annotate other people's observations. So in this case, there's not yet an annotation for the leaves, so I'll go ahead and add that. And in this case, there are no live leaves present yet. Well, that green is on the flower buds. So I'm going to do one more example here. This is an American beech tree that I saw last year. And this time, I'll show you the photos a little bit. There's no photos here that show any kind of fruit or flowers or fruits or seeds or buds. So I'm going to indicate that there. And for leaves, this plant was especially striking because it has both colored leaves as well as green leaves. So I'm going to select both of those. Now, something that you also might encounter is a mistaken annotation on iNaturalist. This is my colleague Allison, who's helping answer questions behind the scenes. She made this observation of a red-berried elder and looks like great clear photos of fruits. So the annotation, whoops, it says flowers here, but I didn't see any flowers. Because this isn't my observation, I can't remove that, but I can disagree with it because it looks like Allison maybe clicked the wrong thing, or this is just a mistake. Mistakes happen on iNaturalist. It's okay. So I'm just going to make a comment here. Kestrel, it looks like you picked the wrong plant phenology. Can you correct it? Leave a nice friendly comment and she'll take a look and fix it so that it is fruits or seeds. And she can remove her flower annotation there. Whoops. So you can do this from individual observation pages like I've just shown, but I also want to show you how you can work through many observations much more quickly. So I've got some filters preset here, but basically what I've done is go to this identify page. It's very useful for identifications, but it is also really useful for annotations. So we're going to drop a link in chat to a similar filtered search. We're just going to focus on the genus Prunus that we already talked about. And I'm going to highlight a couple of other filters here. One is I've focused on the research grade observations because they tend to be a bit clearer. And I only want to, there's this more filter section here. I only want to look at the things that don't have a flowers and fruits annotation already. So now if I update my search, I'll know that all of these are lacking annotations right now. When I click on it, it pulls up this detail view. And across the top here, there's these tabs. We're going to focus on the annotations tab, and we'll be able to move back and forth easily between observations and add annotations. So I don't see any flowers or fruits on this one. And I'm going to start using some keyboard shortcuts to move between these more quickly using my arrow keys on my keyboard. We've also got down here in the left this handy little keyboard shortcuts key, and there's keyboard shortcuts for adding annotations much more quickly, and it'll show you just the ones that are relevant to the species that you're looking at. So I definitely encourage you to check those out. It's much faster than clicking. Here we've got another example with no flowers or fruits. Here we've definitely got some flowers. If I click, I can zoom in a little bit more. Are there buds? It's hard to tell. I know definitely there's flowers, so I'm going to select flowers. I don't see any super clear buds there. Here's another one that's got flowers, and that to me also looks like a bud. It's not open yet, so I will also select flower buds. And you can continue all the way. Oh, yeah, here's a good example of fruits or seeds, and I don't see any flowers or anything else. It's also a good example. Sometimes there's a little bit of a delay, but usually it'll show up if you stay, and even if you move on, it'll show up. Here's another example of fruits or seeds, and here there's a few photos. So I'm gonna check, because there's also in that second photo flowers. And that looks to me like a closed flower bud. So this plant is doing all three things at the same time. Cool. So I'm going to show what it looks like when you go to the end of a page here. It'll show you 30 observations at once. And once you have gotten to the end here, this is a good example of also no leaves, but I'm just, for the time being, I'm just going to focus. I find it helpful to focus on just one thing at a time and go back and separately add leaves. So when you get to the end of your page, it will say you're finished with the page. Do you want to go to the next page, or do you want to mark them all as reviewed? I recommend marking them all as reviewed because then if you've skipped some, you won't be prompted to review them again if you continue trying to you know tackle a manageable number of observations rather than 123,000 of them. So for the next one that I demo, we'll filter to something that feels a little bit more manageable. But I've marked all of these as reviewed. If I refresh the page, I'll be shown totally new observations. For demonstrating leaves, I'm going to move back to the maples genus Acer. And this time I'm going to put in a place filter. So we're working with a more manageable number of observations. We'll filter down just to South Korea. And again, I'm gonna apply my filter to only show me this time the ones that do not have the annotation for leaves. Great. So here we've got a lovely example. Again, we're going to click on annotations here. A lovely example of colored leaves. No green leaves there. Certainly no breaking leaf buds. I think many of you might be also adding annotations at the same time because that took a moment. Here's different colors, but another example of colored leaves. And we should be able to move on even if it hasn't indicated yet. Now here's an example where we've got both green leaves as well as colored leaves. So we'll pick them both. And here we've got just green leaves. And here's a great example of, these are buds, but they're not starting to open. So this is a good example of no live leaves. And I'll show you shortly how that can be really useful in the context of iNaturalist as well as for phenology research. So again, if you get to the end of the page here, you mark this last one, green leaves, and you move on. You've got a much more manageable number of 171 matching observations. So you can mark all of these as reviewed, feel very satisfied. It'll take a few seconds to mark them all that way. And then you can move on and view more unreviewed observations towards perhaps a goal of being able to review all of a particular species in a particular area, maybe for a particular year. All right, so one of the questions that we got was, do cultivated plants matter? In this example, I have filtered just to research grade observations because I thought we would get the clearest examples for demonstration. But if you, for example, are really interested in cultivated plants, you could instead set your filter to casual and specifically ones that are captive or cultivated. And this would show you a set of observations that can still be annotated and can still be used in research, even though they don't have that label. They can still be used in research. And in many parts, there are many researchers who are studying the phenology of cultivated observations. So you can do the same process with different filters. And I definitely encourage you to find an area of interest, a species, and focus in so that it feels manageable and not completely overwhelming. All right, so I want to show you now how adding these annotations can be useful in the context of iNaturalist itself. So this here is what we call a taxon detail page, information about a species. And by default, when you load this page, you see this seasonality chart. This purple passionflower species, one of my personal favorites, you can see that it's almost never observed in January, February, March, November or December, but very commonly observed in the warmer months in the Northern Hemisphere where this is found. So why might that be? If we click on this flowers and fruits button, then it will reload this graph and it will show you the breakdown of observations that have been annotated to different stages. So in this case, no flowers or fruits is shown in green. Red is when there are flowers present. Orange is flower buds. So you can see that flowers and flower buds are often present at the same time. And fruits or seeds, which is this blue line, start to appear especially in August. This gray line is all the observations that are not yet annotated, which is most of them. But there's a handy little filter here where you can hide no annotation, and this chart becomes much clearer. You might even want to also filter this by place. For example, let's see what it looks like when we limit it to Virginia. It'll take just a moment to reload, filtering just to Virginia. And we can look at the pattern of flowers and fruits. Again, we've still got this no annotation hidden. So I definitely encourage you to check out those options. The other reason that these annotations are super useful on iNaturalist is to see what photos look like of different stages. So here I'm switching to common pawpaw, which is a woody plant, because this is a great way, for example, if you want to see what does this plant what do the twigs look like? What does this plant look like when it doesn't have any live leaves? So here's examples of the bark. Oh, it's still filtering for Virginia. So I'm going to remove that place filter, and this is going to give us a lot more. Great, so there's a nice clear photo of what that twig looks like. All right, so in the interest of time, we are right coming up to the hour. I am going to switch back. I have just a few more slides to show you, and couple of reminders that yes, this is being recorded. So later today, we're going to send you an email with a link to this, as well as the links that we've shared in chat. So you have those for future reference. And we would also love to know if you would like to get more emails specifically about phenology and updates about Phenobase. We don't want to send you emails you don't want. So please sign up via the link that we're dropping in chat. We will also put that link in email. I'll just close with the reminder that no one can do everything, but everyone can do something. And I really am so grateful for your time today. And I'm really excited to see what we can do together for plants and for biodiversity more broadly. Thank you all for your time. I'm sorry that I didn't get to all the questions by any stretch of the imagination. Please check out the iNaturalist help documentation and contact us at help@iNaturalist if you have specific questions. And we hope to see you on iNaturalist. Thanks, everyone.